Thank you all. One quick comment, there are even shorter wet-dry cycles than days and seasons. Bruce and I have visited volcanic hydrothermal fields all over the world, really, and there are wet-dry cycles measured in minutes to an hour or so because of hot spring activity, fluctuating levels of water. So just something to keep in mind. We're not stuck with really long daily or seasonal wet-dry cycles. Okay, I gotta give you a few minutes of introduction to get to the unpublished material I want to talk to you about. Green button? Yeah, okay, got it. Okay. So a lot of what I'll be showing you is in fact experimental results, and that leads to a hot spring hypothesis that is testable. The two examples of hydrothermal regions that have been suggested in the literature by ourselves and by the people interested in hydrothermal vents is the so-called black smokers and alkaline vents shown on the left, which in fact are salt water, and the hydrothermal fields that we're talking about, which is distilled fresh water, distilled by evaporation from a global ocean and falling on volcanic land masses. I only have time to talk to you about the fresh water. So the favorable properties that we've observed in fresh water is that organic compounds accumulate on land masses as opposed to being diluted into the ocean. And as Ben had just suggested, they go through a cyclic process, and the deposition is a constant rate. We have found that these pools tend to be acidic, which we think is important for the kind of chemistry we're doing. And the acidity results from SO2, that is a volcanic gas. It turns into SO3, a weak acid, and the pools that we've worked with are all down around pH 2 to pH 3 on the volcanic areas we've been testing. The concentration of ions is very low, millimolar concentration. Wet-dry cycles concentrate potential reactants, and those condensation reactions can then occur in the low water activity. And we've done thermodynamic analysis of this, and it's very clear that there's enough chemical energy produced by the wet-dry cycle to drive ester bond synthesis. Finally, the elevated temperature up in the 80 to 90 degree range provides activation energy for the formation of phosphoester linkages. And last of all, if there are lipid-like components present, the polymers get encapsulated to become what we call protocells. These are steps toward life. They're not life itself, but they are certainly able to evolve, and that's where we are now in our research. So we wanted to test this hypothesis. We fabricated a simulation chamber that puts small glass vials through wet-dry cycles. We control carbon dioxide, elevated temperature, acidic pH range, and the cycles we've talked about. So the device that we've built up depends on this relatively low energy synthesis of ester bonds. And I want to make this clear to you. If you mix ethanol and acetic acid, there's a spontaneous reaction in which water comes off and ethyl acetate is produced. Ethyl acetate is the ester that is part of nail polish remover, if you we're probably all familiar with that smell. However, the same kind of reaction can occur between phosphate and the OH groups on a couple of nucleotides, such as adenosine monophosphate shown here. And if we want to pull the reaction to the right, all we have to do is to have a way for water to leave the reaction so there's no back reaction, and in both cases we should expect to see condensation reactions leading to phosphoester linkages in the bottom thing. Here we have the device that we built up. It's got a source of carbon dioxide, basically keeping out air. Air and oxygen doesn't turn out to be terribly important to us, but we just have CO2 as a way to reduce the possibility of oxidation reactions. There's a heat source that brings up that aluminum disk to about 80 to 90 degrees, our choice of temperature, and that disk rotates about once an hour and brings the vials under a gas flow of carbon dioxide, which dries them down, and then down at the bottom right, you can see a hydration cycle. This is a syringe pump, and the water is dripping into the vials at those 2 points. So at the end of a number of cycles, we will pull the stuff out and analyze it. So I'm going to show you one that we've done with AMP and UMP mixture. We can have a lipid matrix there to promote the reaction. We found that it's favorable and has a protective effect on polymers. We do multiple wet-dry cycles. We bring down the products with ethanol precipitation, a very standard way to bring down polymer products. We do that by spin tube or ethanol precipitation. Then we run either gel electrophoresis or we run nanopore analysis, and you're going to see a little bit of results of that. So the first thing we did 10 years ago now, 2008, with Sudha Rajamani in the lab, was to see whether these polymers could be end-labeled with enzymes that recognize biological RNA. If we've made anything that looks like a biological RNA, these enzymes should at least label the ends of those molecules. And it's the end labeling is done with alkaline phosphatase, T4 kinase, radioactive ATP, and you end up with a radioactive RNA labeled with radioactive phosphate. So this is what Sudha put together. Each of those lanes, these are gel electrophoresis lanes, are separate experiments. It works again and again and again. It's very robust. All of these are different conditions that we put it through. I just want to point out 2 of these. This is the number of cycles. We get more and more and more as we increase the number of cycles. And look at the length of these polymers compared to this ladder here. This is a RNA ladder with known RNA links, and right next to it you can see that these polymers range from 10 mers up to over 100 mers in length. That's really quite extraordinary, that spontaneously we can make polymers long enough to be labeled by these enzymes that recognize AMP. I don't have time to show you all of those, but just a variety of conditions with different lipids and different ratios and so forth. So you can also see these by fluorescent labeling, where we have a dye that binds to a polymer such as RNA. Here we have 2 kinds of RNA that are known RNA. This is a homopolymer called polyadenilic acid. There's another homopolymer, that's polyadenilic acid, poly-A. Notice that the dye, which is ethidium, an intercalating dye, does not strongly label these single polymers. But if we make a mixture where we get, in fact, a double helical form of a poly-A, poly-U duplex structure, now these microgram quantities are labeled by the dye because the dye intercalates between the stacked bases and becomes fluorescent. And here is our polymer product, again ranging from 20 mers up to the 100 mer range. Here's another dye called CyberSafe, and here's our product. Here's a DS double-stranded 20 mer right there, and here's our product. So we were pretty sure that we were making something that was a polymer, but we wanted to try one more way to look at this. So we did what is called nanopore sequencing. Now this is new, so this hasn't been published yet. We're repeating this. These are sort of preliminary work I'm going to show you now. So the idea of nanopore sequencing is that we can lead a double strand of DNA or RNA, in fact, into a molecular motor which ratchets the DNA bases at about 450 nucleotides per second through a limiting aperture in a protein nanopore. That aperture has about 3 bases in it at any given time. As the bases are ratcheted through that area, we have a change in the electrical current in the picoamp range, which reflects the base sequence. So this is now out as a commercial device. It's called the MinION. I'm going to show you one of those, and also the Prometheon. This is Oxford nanopore technology, building on the nanopore idea. So let's take a look at the next slide. Here is what I'm going to show you. We wanted to be sure that we really could make something that was exactly the same as a strand of DNA. So we kept it simple. We decided to use TMP, thymidine monophosphate, as the monomer. This can only make 3,5 phosphoester linkages. We put it through multiple wet-dry cycles. We expect to get oligothymidylic acid, just a whole bunch of T's in a row, and then we can isolate that. We ligate it to a known strand of DNA in order to differentiate between the signal we're going to get from the oligo T and a known base sequence of DNA. And then we put that through nanopore sequencing. So here's what it looks like to use the MinION. Your sample is injected into this handheld device. It goes to 2,000 nanopores in this little area here. That's the sensor chip. Each one is independently being referenced by the electronics of the system. All of the electronics are in this device, and the signal is fed into your laptop computer. This has gone all over the world now, looking for Ebola in Africa. Every 90 minutes, it goes over our heads in the International Space Station. The MinION is being used to sequence the DNA of the organisms that now coat the interior of the ISS. So let's take a look now at what we see. What we see as a single nucleic acid going through the pore looks like this. This is the known sequence going through, and the up and down is, in fact, the AGCT of that known bit of DNA. And then right there is this long string, which is just the what we think is the oligo T homopolymer passing through the pore. We have a base calling device that allows us to turn this electrical signal into the nucleobases, and this is what it looks like. Here's a gel of the radioactively labeled oligothymidines, starting down around 30 mers, going all the way up to about 90 mers. You can see these individual oligothymidines showing up in this gel, and here are the base called fragments. The oligo T we see again and again attached to the known RNA ligated to the known I, and this then is the oligo T now attached that we have synthesized. This at least convinces us that we can make very long strands of nucleic acids using just wet-dry cycles. The 30 mer, by the way, is down in this range. You can see that these links match what we see here. This is an intermediate length, approximately in the 40 mer range. And then here is a very long one up in the 80 mer range. Okay, now what we want to do is to see whether this actually works out in the wild. Oh, I want to show you this too. This stuff accumulates inside the lipid vesicles. If we have a lipid there, we can stain it with acridine orange. This is starting with AMP and UMP. These are the lipids and the encapsulated material after about 3 cycles. Here is with DNA. This is DAMP and TMP, and you can see the DAPI now staining these vesicles with this encapsulated DNA. So we're pretty sure that not only can we make it, but it becomes encapsulated to make protocells. Now we're going to take it out into the wild and ask this last question of my talk. Does it actually work in conditions such as we assume were available on the early Earth? What you just saw is a hot spring in Rotorua. Bruce was down there just last summer doing this work, so he's my co-author on this talk. He would put these tubes in a large aluminum plate. There's approximately 100 of them. they have the dried material that I did in the lab at UC Santa Cruz. And then he put these through 4 cycles, about 30, 45 minutes each, and he added water from an acidic hot spring for the recycling, so 4 cycles. Here is the products that we're getting out. Notice that the products containing U were more abundant than the products containing A, which was interesting. We didn't expect that, but that's just the way it turned out. We then did a gel of this and compared what I showed you in that first gel with the products. Here's what I showed you the first time around, and here is what we see in the hot spring cycles. So we're convinced that this is not just a laboratory phenomenon, but also can occur out there in the real world. And I recommend that people doing work in the laboratory, once in a while, step out of the lab into a place that we consider to be a prebiotic analog environment and see if your result actually works under real conditions. So finally, I'll end up with our conclusions. This is an alternative scenario that we're developing for the origin of cellular life. We think that life began in freshwater hydrothermal pools subject to cycles. Life did not invent nucleic acids. Instead, life discovered pre-existing hydrothermally cycled polymers and encapsulated them to make protocells. If amphiphiles are present, they get encapsulated. If you have protocell populations, they vary, just as is always required for evolution, selection and evolution. There had to be variables. They can undergo selection and evolution. And what we're now looking for is how to test this conjecture that life began when rare systems of encapsulated polymers happen to be capable of the catalytic functions related to the life process. We put this into a sort of a geological scenario, and that's just my last slide now. And we have a synthesis of organic material being accumulated on these volcanic land masses. As these pools go through cycles, the material gets concentrated and begins to react. Here is the reaction cycle. You can see going around this circle between a wet phase, down drying to a dry phase, and back to a wet phase. This cycle then builds up, cycle after cycle, increasingly complex protocells. As they accumulate, they get distributed downhill toward the seawater ocean and finally adapt to seawater, where they colonize the ocean as the last step in this origin of life scenario. Thank you.
I'm afraid we're a little bit limited for time, but please find Dr. Deamer if you have any questions for him, and we'll go to our next speaker, Vincent Ridgie, please. Thank you, Dr. Deamer.
Okay.